Hello biologists, welcome to your first concept of Unit 3 Biology. We are going to be recapping some of your knowledge of Unit 1 and 2 and also build upon some of the concepts of the cell membrane and transport through it. Hopefully you will be able to have a general understanding about this already and excel through Head Start. We are going to assume that you have a basic understanding of the role that most of the main organelles of the cell play already. Though we will touch upon these, if you aren't too familiar with it, then check your textbooks as a start. The plasma membrane is our main focus. It's sometimes called the cell membrane, the differentially or selectively permeable membrane. This is the barrier between intracellular and extracellular areas. Inside of this is the cytosol. It's the fluid within the cell. You can also find the cytoplasm here, which is the fluid within the cell and the organelles as well. The main role of the plasma membrane is to enable substances that the cell wants to move into and out of it. This is an important process as waste and nutrients flow through and are controlled by the membrane. The plasma membrane also encloses most of the organelles in eukaryotic cells, as you can see some examples here, and it also allows for some communication and recognition between cells to occur. The structure of the plasma membrane is made up of molecules called phospholipids, and the way they interact and are positioned within the membrane is known as a phospholipid bilayer. The bilayer has a circular phosphate molecule as the head and two fatty acid tails. The layout of this bilayer is very important as the hydrophilic or water-loving heads face the outside of the bilayer and the hydrophobic or water-hating fatty acid tails face inwards to each other, creating a lipid and oily layer in between. The structure of these phospholipids play an important role in the transport of molecules, so it's important we have an understanding of this. Other structures that we can find on a plasma membrane are integral proteins. Examples are carrier and channel proteins. They penetrate and sit through the whole of the bilayer from one side to the other. The role of this is aiding transport across the membrane for different processes. Peripheral proteins sit in the surface of the membrane and mostly play a role in communication. Cholesterol is located between the phospholipids of the bilayer, and it helps maintain some consistency within the membrane. And the carbohydrates, they attach to proteins in the membrane. We often call these glycoproteins, and they play a role in cell recognition. The fluid mosaic model is an important concept for you to understand, and often something that can appear on assessment. Essentially, it is a description of the phospholipid bilayer in terms of structure. You can see here from this diagram, we've got those red heads of the hydrophilic molecules that we were speaking about before. The two fatty acid tails here are pictured in green, and you can see that the fatty acid tails are almost touching each other, playing a little game of footsies in the middle of that bilayer there where the fatty acid environment is. And you can see that the red hydrophilic heads are opposite each other in the water environment or the hydrophilic environment. Essentially, the fluid aspect refers to the ability of the phospholipids to move laterally and in most directions where needed, and create this almost bubble-like structure that can rotate around itself and always move. This makes the membrane very flexible, and it means that it can be very hard to break and puncture, as you can often see through little clips of IVF on the news sometimes, where the egg cell is penetrated by a needle. When the needle is removed again, the membrane returns to the original shape. The term mosaic refers to the pattern of the molecules stuck together, and brings us back to the last slide where we were discussing not only the phospholipids, but also the different types of proteins that are found here. Now that you have a refreshed view of the structure of the plasma membrane, we can now look at it in action and examine the different processes of where molecules will move in and out of the cell. Diffusion is the main process of movement of molecules Osmosis is a type of a diffusion when the movement of a solute, such as water, occurs moving through the membrane. Facilitated diffusion requires a peripheral protein, such as a channel protein, and active transport is the movement of molecules against a concentration gradient requiring energy. It is the only process out of those first four that require energy, making it a little bit different. And endocytosis and exocytosis are what we call bulk transport, when having large molecules or a collection of molecules exported out of a cell or entered into it. When describing these processes, it's really important to use really detailed language here and practice your phrasing. Diffusion is the movement of solutes from a high to low concentration down a concentration gradient. Diffusion will continue to occur until equilibrium and equal concentrations of these particles are both in and outside the membrane. And though particles never stop moving, the concentration will stay equal once equilibrium has been reached. This is a passive process, meaning it doesn't require energy. And the best example of this in a non-living system, such as a classroom, would be one that has been sprayed with deodorant or an air freshener. These particles will move through the room from an area of the spray until the spray has equal strength all around the room. We can speed up the process of diffusion by using smaller particles. We can increase the amount of the concentration of the particles. We can increase the temperature, or we can use a gaseous medium to dispose of the particles, like in air. It is important you can understand this, as this is a key process in terms of altering experiments or altering this process. The chemical structure of the molecules that we see moving through a plasma membrane play an important part as to the process they follow to cross the membrane. Small non-polar molecules we will be able to pass directly through the membrane without the assistance of a protein molecule and between the phospholipids because of their size and their charge. As a molecule such as water is small, it can still diffuse through the membrane due to its size, even though the polarity of water means the process is slower than it would be if a non-polar molecule like oxygen or carbon dioxide were to try and move through that membrane. Osmosis, in comparison to diffusion, is still an example of diffusion. However, as the molecules that the water is mixed with, or the solvent, are often too large to cross between the phospholipids of the membrane and diffuse easily, the solvent will move across the membrane, balancing the concentrations of solute and solvent. The movement is from an area of low solute to high solute concentration across a semi-permeable membrane. Once again, after equilibrium has been reached, molecules still continue to move. However, the concentration will stay equal. Due to the solution being watery and aqueous, the movement is slower than if it were to occur in a gaseous medium. Osmosis is a passive process and does not require energy. Examining some examples of osmosis occurring here and situations where we examine osmosis, if the cellular environment inside the cell and the external environment where the cell has been placed has the same concentration of solute, then the solutions are termed as isotonic, and water molecules will not need to cross the membrane as concentrations are already balanced. If the cellular environment has a lower concentration of solutes than the extracellular environment, then the cell is said to be placed in a hypertonic solution, and movement of water would occur from the intracellular environment, where there would be low concentration of solute, to the high concentration of solute area, which is outside of the cell, to balance the concentrations of solute. This will cause the cell to shrivel. If the cellular environment has a higher concentration of solutes than the extracellular environment, then the cell is said to be placed in a hypotonic solution, and movement of water would occur from the extracellular environment, where there is low concentration of solute into the high solute concentration area inside the cell to balance the concentrations of the solute. This will cause the cell to swell and potentially it might burst. Here we can see the outcomes of the cells we were just discussing. The yellow cells above depict the results of an animal cell, and you can see that when placed in a hypotonic solution on the left there, the cell could potentially burst open and lyse. While in the hypertonic solution on the right hand side, the cell shriveled up and got smaller as all the water rushed out of the cell. In the plant cells below that, the cells in the hypotonic solution on the left were protected by the cell wall, and though they swelled and did not burst, they did become what we call turgid or slightly enlarged. The cells in the hypertonic solutions on the right, they shrunk away from the cell wall and they underwent what we call plasmolysis, or they plasmolysed. If you have completed your egg demonstration already, you can start to think about this theory and how this was reflected with the eggs. Facilitated diffusion is another type of diffusion. In this case, the solute molecules are moving from a region of high concentration to a region of low concentration, just like in normal diffusion, and they still cross the semi-permeable membrane. The difference this time is that the process requires a transport protein, because the particles may be too large or they might be polar, and therefore not able to fit between the phospholipids. Channel proteins or carrier proteins will be employed to assist this process. Here we can see an example where channel proteins are used when a water-soluble ion is too large to pass between phospholipids, and instead passes through the membrane from a region of high to low solute concentrations. As the channel proteins move apart to allow a space for molecules to pass down the hydrophilic or water-loving lining, the phospholipids are able to move sideways, demonstrating the fluidity of the membrane that we spoke about previously. As particles are moving from a high to low concentration, this is a passive process and does not require energy. If we compare the channel proteins on the previous slide with the carrier proteins we can currently see on this diagram, we can see that carrier proteins require a specific molecule to bind to the protein. The protein will then change shape and deposit the molecule onto the other side of the membrane. This term specific is so important throughout the whole year, so make sure that you use it and reflect on it in other concepts as well. Once again, molecules are passively moving from high to low concentration. The last example of diffusion, or a type of diffusion, is active transport. And this is a process that is largely different to the previous three, as it is an active process and molecules are passing through the plasma membrane from a region of low solute concentration to high solute concentration, and they're moving against the concentration gradient. This requires energy, or ATP, and this is a very important process in depositing waste from a cell or taking nutrients into it. Cells undertaking this process at a high rate will require a large number of mitochondria to provide the ATP for this process to continue functioning. In comparison to the diffusion processes we've just spoken about, larger molecules can still be transported across the plasma membrane, but they use this process called bulk transport. The first example of bulk transport is exocytosis, and this is used when particles or molecules are exiting the membrane. The vesicles that these particles are held in will fuse within the side will fuse with the plasma membrane, and the vesicle contents are then released from the cell. This requires the phospholipids of the plasma membrane to once again move laterally and allow a joining of these phospholipids from the vesicle, again moving back to that fluidity of the membrane. The second type of bulk transport is endocytosis, or when particles are entering the cell. Through endocytosis, or bulk transport into cells, this happens when part of the plasma membrane will form around particles or a molecule and create a vesicle. So the phospholipids will break off from the plasma membrane, form a small vesicle, and then allow those molecules to move throughout the cell to the area needing to go. The most important example of bulk transport is the exocytosis of proteins. And we'll speak a little bit more, actually a lot more, about this process in more detail at the start of next year. But this example is showing where the proteins are made through the ribosomes, often on the rough endoplasmic reticulum. They're packaged into a vesicle and sent to the Golgi complex in the middle there, where final alterations are made. Then these secretory vessels will move to the plasma membrane, and they will allow this vesicle to open up, join with the other phospholipids of the membrane, and discharge those particles or these proteins that we need to move outward out of the cell.